The topic for our groups today is enzyme, induced-fit models and Michaelis-Menton's kinetics. Question number 12. In the induced-fit models of enzyme actions, the enzyme active site blank. Possible answers. A. Stay the same shape while causing a change in the shape of the substrate. B. Adjust shape to adapt to the shape of the substrate. C. Uses a cofactor to change the shape of the substrate. D. Uses an inhibitor to adjust its shapes for the substrate. E. Stays the same shape during substrate binding.
An enzyme is a type of protein or a molecule that is made up of proteins that help the substrate for it to turn into products. The induced-fit model is a proposed mechanism of interaction between enzyme and a substrate. It postulates that the exposure of enzyme to a substrate causes the active site of enzyme to change its shape in order to allow the enzyme and substrate to bind, and eventually the substrate will turn to products during that process. And we're going to talk about this in more in detail as we go over these answer choices.
Option A. Stays the same shape while causing a change in the shape of the substrate. This option is incorrect for the induced-fit model of enzyme actions because the structure of the enzyme has flexibility to adjust the shapes of the active sites for binding different substrates, while the substrate can only slightly change its shapes. Moreover, the substrate does so only to fit the newly modified active sites in the enzymes in order to increase the range of the variety of the substrates.
The answer is B because the active site of the enzyme has flexibility which will allow for the shape to adapt according to that of the substrate. Furthermore, the substrate may also modify itself to better fit the newly formed geometry of the enzyme's active site, which results in what we call the dynamic model of enzyme action or the induced-fit model. The end result is that the substrate and enzyme can work together to fit an optimal geometric arrangement, and this allows for a lower activation energy for the reaction to occur. The answer is not C because the enzyme does not require this small additional non-protein molecule known as a cofactor in order for it to fully function and achieve the desired and optimal shape at the active site or the substrate. Although the cofactor can perform other tasks that the enzyme needs, such as the increasing rate of catalyzing substrates.
The answer choice D states that in the induced-fit model of enzyme action, the enzyme active site uses an inhibitor to adjust its shape for the substrate, and I have to say that it's not true because when the enzyme binds to the substrate, they by themselves act as two pieces of clay and mold together to make a perfect fit, and that is basically what an induced-fit model of enzyme action is. And inhibitor does not play a role, and it does not contribute to the induced-fit model of enzyme action. Instead, an enzyme inhibitor binds to the enzyme and decreases its activity by changing the active site. Therefore, D is not a correct answer. The answer choice E states that in the induced-fit model of enzyme action, the enzyme's active site stays the same shape during substrate binding, and it is not true because when enzyme recognizes and binds to the specific substrate in the process called initial binding, although they are attached together, the force of holding these together is not strong enough. So what happens is that the enzyme and substrate both change its shape a little bit until they are perfectly bound to each other, and This process is called induced-fit model of enzyme.
To answer question number 13 relating to the topic of enzyme kinetics, we should first understand what Michaelis-Menton equation is. Michaelis-Menton equation states that it measures velocity of a reaction with increasing substrate concentrations while keeping pH and temperature at optimum. When you plot reaction velocity against substrate, you will see that enzymes that follow Michaelis-Menton kinetics show a hyperbolic curve, while allosteric enzymes show a sigmoid curve. In addition, when you look at the figures, you can see that at Vmax over 2, the substrate concentration is Km. So the answer would be a graph of initial velocity versus substrate concentration will be a hyperbolic curve, such that Km is equal to Vmax over 2, and as the substrate increases, the initial velocity will eventually approach Vmax.